Bacteria are the smallest and most numerous organisms in the world, which means that overall they're smaller than any other living thing, and there are more of them in the world than any other living thing. So you could say they're the most successful organisms because they've reproduced more than anything else in the world. There are two types of bacteria, two kingdoms that include bacteria, and they are eubacteria and archaebacteria. Eubacteria is known as the true bacteria because it's the common bacteria that you think of when you think of things that live in your digestive system or things that might get on surfaces that have to be cleaned, things like that. That's going to be eubacteria. It's found in common places. Eubacteria also has cell walls made of peptidoglycan, which is a chemical. It's a polysaccharide that makes up those cell walls. Now, archaebacteria has cell walls still, but their cell walls are made of other polysaccharides, and they can be made of a lot of different things, but they're not made of peptidoglycan, so that's one of the main differences what their cell wall is made of. Archaebacteria is also going to be found in very extreme environments. That's why they're called extremophiles. Phile means love, and so they love extreme environments. Things that these two kingdoms have in common are that they are prokaryotic, which means they have no nucleus. They instead have a nucleoid region that has no membrane around it, so it's not actually a nucleus, but they still have DNA. They also have cell walls, even if they're made of different things, and they're always going to be unicellular. There's no such thing as a multicellular bacteria. They'll always be only made of one cell. So let's look at some different kinds of archaebacteria. Archaebacteria can be one of three things, or commonly one of three things. We look at methanogens last unit. Methanogens' job is to take carbon dioxide and hydrogen, and through a chemical reaction they convert it into methane. And methane is kind of a smelly gas. And in cows' digestive systems, there are these methanogen bacteria that digest the cellulose that they eat, the cellulose in the grass. And so cows actually release the methane that's created in this chemical reaction. They release up to 300 liters of methane a day, which is a lot. And they release that through either burping or passing gas. There are also thermophiles. These are bacteria that live at very high temperatures, places like hydrothermal vents at the bottom of the ocean, temperatures where most things would not be able to survive. And then there are also halophiles like salty environments. And so they grow in very salty places like the Great Salt Lake. And usually in places where these are found, the water is almost red because they're sort of red in color. This is a picture of a typical bacteria, and you can see that in this bacteria there is DNA, but there's no membrane around it to make a nucleus, and so it's just the DNA floating freely. Notice it has ribosomes because it needs to be able to make protein, so it has ribosomes to make the protein that it needs. It has a cell membrane and a cell wall, and this one happens to be a eubacteria with peptidoglycan in it. And in addition to those things, you might also find some flagellum or cilia that help the bacteria move around. And those are the basic parts of a bacteria. Alright, most bacteria are heterotrophic, which means that they have to eat food to survive. They get food from other organisms. However, some bacteria are autotrophic. And if they're autotrophic, that means they have to produce their own food. They make their own food. There's two processes that they can do to create their own food. They can use photosynthesis, where they use sunlight as a source of energy, and in the process of photosynthesis they make glucose, which is their food. Or they can use chemosynthesis, which is similar to photosynthesis, but instead of using light, they use chemical reactions and the energy produced in the chemical reactions to make glucose or some other form of food. Now, bacteria have a sort of system of naming that they follow based on their shape and how many of them there are found at a time. So we'll look at some pictures over here of bacteria. We've got some that are round and spherical. Their name will have coccus at the end, or in plural it would be cocci. If they're sort of rod-shaped like a pill, then they're going to have bacillus at the end of the name, or in plural bacilli. And if they're sort of spiral, kind of snake-like, they call this helical. They'll have spirillum at the end of the name, or spirilia in plural. So the end of the bacteria's name comes from its shape. And then the prefixes, the very beginning of the name, comes from how many there are at a time. So if you find two bacteria together that kind of hang out together in pairs, it might be diplo, and so you would pair that with whatever name it is. If it was two round bacteria, it would be called Diplococcus. If there were four of them together, it would be tetra at the beginning of its name. If there's a cluster of more than four, it would be Staphylo. The bacteria that causes a staph infection is actually a type of Staphylococcus. And if it's a chain of bacteria, not a cluster, then it would be Strepto. The bacteria that causes strep throat is a type of Streptobacteria. And so the names kind of come from the shape and how many of them there are. Now, bacteria can reproduce very rapidly. In fact, a bacteria in a good environment can reproduce up to every 20 minutes. And so that's if it has a warm and moist environment. And that would produce, it would double bacteria in numbers every 20 minutes. That's very, very rapid. But the thing that stops bacteria from constantly reproducing, because if it reproduced at this rate constantly, we would be overrun by bacteria. But because there's a lack of resources in a lot of the environments where these bacteria live, they can't reproduce that rapidly, and so a lack of resources keeps them from reproducing. So that's like a lack of food, a lack of the correct temperature or pH, and so it prevents them from reproducing. There are two main types of reproduction that bacteria go through. Both of them are asexual, which means they don't have a male and a female role. They're just the one bacteria doing it on its own. And so the first type is binary fission, and in this is kind of like the typical simplest form of reproduction, where the genetic material, the DNA in the bacteria, is copied, and it goes to either side of the cell, and then the cell divides in two. It's very similar to mitosis, but it's actually much simpler. And then the two daughter cells that are produced will be identical to the parent, so there's no genetic variation unless there's a mutation that happens. The second type of reproduction is conjugation. This is also a type of asexual reproduction, but it's kind of similar to sexual reproduction because it does produce some variety. So what happens during conjugation is two bacteria cells will get really close to each other, and a protein bridge will form between them. And that protein bridge is called a pilus. And what will happen is the genetic material of each bacteria will move over the pilus, the protein bridge, and they'll switch genetic material. So they get some variation in their genetics, and so this gives us sort of a variety in the types of bacteria that we have. Now, sometimes a bacteria will face a time when they don't have everything they need to reproduce, and so they'll have to find a way to deal with that. One of the ways they deal with that is by forming spores. So if they're faced with unfavorable conditions, they can actually make sort of an envelope around them called an endospore. And that endospore will contain their DNA, and the DNA will be surrounded by this thick outer coating. And then it'll just pretty much go dormant for as long as it takes for the conditions to get better, so that it can survive for years under these conditions. But later, when the conditions are better, when there's the right temperature or pH or enough food for this bacteria to survive, the endospore will sort of dissolve and the cell will begin to divide and reproduce again. So bacteria are actually very resilient. They can survive a lot. There's a lot of important uses for bacteria. We find bacteria important for digestion, for food production, like in yogurt. You have to have bacteria culture to make yogurt. It's important in medicine production, in things like antibiotics. It's important for ecological decomposers, so that we're not overrun with trash. Bacteria have to decompose a lot of waste that we produce. And they can cause disease, which is a pretty important role that they play. They cause a lot of different diseases, but not all bacteria cause disease. Not all of them are bad. There's a lot of ways that we use to deal with bacteria, at least the harmful bacteria that can cause disease. We can use antiseptics. Antiseptics are used to kill bacteria or microorganisms of any kind that are on living tissue. So you can think about like rubbing alcohol and lots of different kind of antiseptic sprays. Disinfectants are used to kill microorganisms that are on non-living surfaces. So things like Lysol and other things that advertise that they kill microorganisms are disinfectants, not meant to be used on living tissue like skin. And then the other one is antibiotics. And antibiotics is specific to bacteria. And there are certain antibiotics that work better for certain types of bacteria. And what it does is it either kills or slows the growth of bacteria. This is the most effective way to fight bacteria, especially in the human body. So antibiotics are actually not just given to humans. A large portion of the antibiotics that are prescribed and given out in the United States are actually given to animals. And specifically cattle, because they're susceptible to disease as well. And this has led to a lot of new strains of bacteria being formed in our meat supply because all these antibiotics are being given to the cattle.